sorry. Okay, can everybody hear me all right? Awesome. So thanks, Roy, for that introduction. And I'm really excited to be here tonight and to have the opportunity to tell you all about my favorite topic in science, which is the microbiome. And hopefully by the end of the night, I'll have really imparted upon you the complexity of your microbiome and how really important it is for your everyday and long-term health. So, what are you going to learn here tonight? What should you go home knowing after you've talked with me? Hopefully, you realize that your microbiota plays a very intimate role in your everyday and long-term health, but that this relationship that you have between you and your microbial pets that are all over you is actually a really complicated one, and it's difficult sometimes to discern whether your microbes are doing good or bad things for you. And we'll use my own work as an example to get some of these ideas across to you, which is I study the infant gut microbiome and how these microbes that are associated with infants are actually really, really important for stimulating the development of these insulin-producing beta cells that are in your pancreas. And we'll talk more about those cells as the night goes on. And we'll talk about how harnessing these infant microbes poses both risks and rewards, but how my lab is trying to navigate these challenges in order to utilize them to treat diseases like type 1 diabetes. So, your microbiota. This is the star of the show tonight. Your microbiota is the collection of all of the microscopic organisms that are inhabiting your body. And so this includes things like bacteria, viruses, microscopic fungi like yeast, as well as other small, creepy, crawly eukaryotes. And the truth is that you guys, sitting here in this audience right now, all these microbes are crawling all over you, okay? Yeah. And you actually have an equivalent number of microbial cells on you right now as you do human cells. So for every human cell in your body, you have one microbe. In fact, you have 10 times as many microbial genes as you do human genes. So we could argue that you're more microbial than you are human, even, which is maybe a little bit unsettling. And the majority of the microbes that live on you, they actually live inside of you. They live inside of your gastrointestinal tract or your gut. And there's so many of them in there that they account for about 4 pounds of your total body weight. So next time you get on the scale and you're worried about the number, subtract 4. That's your microbes, okay? But that means that this room's not quite full, but if it was full, that would be about 800 pounds of gut microbes just sitting in this room right now. Maybe gross, maybe cool, depending on who you are. And within that 4 pounds of gut microbes on your body, there's anywhere from 500 to 5,000 different species of bacteria alone. And that number between 500 and 5,000 is really dependent on the individual. So we each have individual differences across our microbiome. And remember that number because we will come back to it a little bit later. So I don't want you, though, sitting there getting the impression that your microbiome is just this 4-pound bag of sludge that's traveling through your gastrointestinal tract. Because it turns out that that's not at all the case, okay? So we know that because as microbiome scientists, we can actually see the microbiota. We can see it in real time. How do we do that? Well, we have some very cool model organisms that actually allow us to visualize the microbiota. So this is not the microbiota. This is a fish. Many of you might recognize this fish. It's a common member of your local pet store, and some of you might even have aquariums at home that have this fish. It's a zebrafish. The Latin name for it is Danio rario. But the zebrafish, it turns out, is a powerhouse of developmental biology because as a baby zebrafish, or a larval zebrafish pictured over here, They're transparent, so we can actually look into this fish and visualize all its internal organs, which is great for microbiome science because we can look inside of its gut, inside of its gastrointestinal tract. So this yellow arrow here is just highlighting the zebrafish gut, and you can see right into it, which is very, very cool. And this is basically the fishy equivalent of your own intestine, of your own gut, okay? And so if we do that, if we look inside this zebrafish's gut, we can watch the microbiome. So what you're looking at here in black, it's a little bit hard to see maybe on this white background, but this is the zebrafish's intestinal cells. And this video, throughout the 6 seconds that it loops, you can hopefully see these cells contracting and peristalsing, which is something your own gut does. But zooming around inside of that gut, like little tiny bees, are actual bacteria. And you're watching this video happen in real time. So each one of these little dots is an actual microbe zipping around inside of that zebrafish gut. And what I want to impart to you is that this is not a pile of sludge that's just traveling through you. This is a highly dynamic and interactive part of your body. And each one of these bacteria is not only interacting with the other bacteria in there, but they're secreting all kinds of products that are also impacting your own biology. So they're really important. And these guys probably look like they're really at home in there, right? They look like they belong. That's where they live. And that shouldn't be that surprising because they do belong. We evolved in a world that was dominated by microbial life. So if we rewind all the way back to the very, very beginning, when Earth was born, approximately 4.5 billion years ago by our best guesses, right? It did not take very long after Earth was born for the first life to form, and this was microbial life. It only took 1 billion years, which in the grand scheme of all of this is really not that much time. But microbes were around for a really, really, really long time before the first multicellular life actually formed, which was about 600 million years ago, which to us seems like a long time. But microbes were around for this whole event and probably stimulated some of it happening even. But humans, we're just a tiny blip on this radar. We've only been here for 2 million years. And if we think about the time that modern humanity has been trying to harness biochemistry to interact with us medically, to prevent medical illness, it's like an even tinier blip on that tiny dot, right? But microbes have been interacting with multicellular life biochemically for 600 million years, so they've probably figured out lots of ways to impact multicellular life that we haven't thought of yet. And so what this means as microbiome scientists is that this is a really exciting well of potential novel biochemical activity that we could harness to treat human diseases, to interact with our own cells, right? And so we're so excited about this that we've put a lot of effort recently into understanding how microbes impact animal life. How do they impact humans? And we've learned a lot from this. This is a really complicated figure, all right? And I don't expect you to sit there and read through this figure. But what I'm trying to show you is that we've barely been studying this process, and this one figure, which is something I was commissioned to put together a few years ago, summarizes all the ways that microbes impact human health. And it's really, really complicated because our relationship with microbes is incredibly complex, and it turns out that they can impact every single biological system on and inside of you, from your gut, where they help you digest nutrients and produce important vitamins, to your brain, where there aren't even any microbes present, but they're still controlling things like your mood, and they even help you decide what you want to eat. They even send you craving signals. It's kind of creepy and weird, right? And so in our attempt to summarize all of these things onto one page, which is really a futile effort, we also tried to impart the fact that all of these interactions, not all of them are good. Some of them can be detrimental. And what we're learning as microbiota scientists is that whether something is good or bad for you is really context specific. And it really depends on the individual's own biology, okay? And so what defines a microbe as one of these things or the other? How do we know if it's something good enough to put into our fermented drink and drink down and get some kind of health benefit, or whether it's something that's going to kill us, right? It turns out that this relationship is not always so clear. And the same microbe can actually be both. It can be pathogenic and life-threatening, or it can be commensal and doing really important things for your health. And so to demonstrate this, I have one microbe to make an example of, and this is the microscopic yeast Candida. And Candida comes in two forms. It comes in this pathogenic form that forms these long, stringy, hyphal-like structures, and it comes in this commensal form that doesn't have those nasty hyphae that are sticky and invasive. And in its pathogenic form, it really causes life-threatening systemic infections. It can be a really dangerous microbe. It also causes localized infections that you guys might know of as athlete's foot, pesky nail fungus, and thrush, just to name a few. But in its commensal form, it actually does really good things like shape your immune system and provide protection from bacterial blood infections. That's really powerful stuff. And as I'll highlight later on in the talk, this microbe also promotes growth of your insulin-producing beta cells. So it's hard to decide what is the nuance here between a pathogen and a commensal. And I think as humans, we have a tendency to have a negative interaction, something that causes us harm, and that harm is so great that that microbe is forever labeled a pathogen. And the risk of that pathogen is too high for us to even imagine the good that it could possibly do, right? And so to illustrate this point, historically, humans have a pretty dicey history with microbes. They've caused really devastating and terrible diseases. And so in the background here, this is a famous painting called The Triumph of Death, and it's depicting the European Black Plague that happened in the mid-1300s. This is a illness that was transmitted by a bacteria called Yersinia, okay? And so it's fair to say that humans have encountered a lot of really, really terrible pathogens in our history, right? And then we probably have a bit of PTSD from events like this that's ingrained in us, all right? And over time, we've really learned to understand microbiology, and we've harnessed a lot of tools to help us prevent these terrible, terrible infections, okay? So we've developed things like vaccines and antibiotics that keep us from being infected and harmed by these pathogenic microbes. We've also developed ways to distance ourselves from these terrible microbial exposures, right? Don't even let the microbe get to us in the first place. So we have things like plastics and refrigeration. And antimicrobial cleaners are a common household product, right? We all use these in our homes. We use them at the doctor's office. And all of these things combined have really led to the eradication of a lot of these pathogenic infections in a lot of places in the world, which is not a bad thing, right? But something else has also been happening alongside of the eradication of these diseases. And that is the new modern diseases have been rising. Autoimmune diseases like Crohn's disease, multiple sclerosis, type 1 diabetes, and asthma. And these are diseases that were much, much, much rarer, and they have been expanding in incidence, coincidentally with the decline of a lot of these infectious diseases, okay? And so one theory that people have been proposing kind of over and over in the last couple of decades is that perhaps in our effort to distance ourselves from these terrible pathogens, we've also distanced ourselves from some really good and helpful microbes that may have been preventing some of these diseases, okay? And so this is called the disappearing microbiota hypothesis. It builds on things you've probably heard of before, like the hygiene hypothesis. And this idea was most recently put forth back in 2009 by two very well-known microbiologists, Stan Falco and Martin Blaser, pictured here. And the idea is that in our effort to distance ourselves from microbes and keep ourselves clean, we may have overdone it just a little bit, right? Maybe those antibiotics that wiped out that pathogen also had collateral damage and wiped out a lot of the good things, right? And so maybe that's actually causing us harm that we haven't thought of before. So how do we test this idea that fewer microbes are actually causing disease? Well, we can actually sequence microbes associated with health and disease, which is a really powerful tool that microbiologists haven't always had. And so in the early 2000s, the NIH started this project called the Human Microbiome Project. And the goal was to sequence all the microbes. What are the microbes associated with humans in both health and cases of disease? And the way that we went about this was to sequence one gene that all bacteria have, called the 16S gene, okay? And each bacteria has a different variation of this gene. And what it allows us to do is to understand not only what microbes are present, so not only who's there, but how abundant is each of those microbes in a given sample. Okay, so this is a lot of information if we're trying to understand how someone with a disease might have different microbes from someone that's healthy. And to just take a small aside, the University of Colorado Boulder actually has some history and a hand in the Human Microbiome Project. And so there's a former faculty member who was one of the earliest hires in BioFrontiers, actually, Rob Knight, who played a big role in helping us develop the tools to analyze 16S sequencing data from humans, which is really awesome that we have this history here. And so Rob Knight and friends at the Human Microbiome Project, they did this 16S sequencing on all kinds of people around the country, people with disease and people who were healthy. And what they kept finding over and over and over again was that a lot of patients with some of these diseases we've been talking about actually have fewer species of microbes in their microbiome compared to people who are healthy. So maybe someone who is healthy has 5,000 species in their gut, but someone who has these diseases might have as few as 500, okay? So really this showed that a lot of these diseases on this graph, like Crohn's, which is an inflammatory disorder of your intestinal tract, these people have fewer different types of microbes in their gut. And it turns out that this same observation is true for type 1 diabetes, which is the disease that my lab studies. And so we're very interested in how the loss of microbes contributes to this disease. So diabetes is a global health problem, and it's only predicted to get worse. So this is a map of the world from the International Diabetes Federation. It's very up to date, and it illustrates the rate of diabetes in every country. And what you should notice first is that there is no country that doesn't have diabetes, first of all. It's a really prevalent disease. And all of the places in red are places with the highest rates of diabetes, okay? And what you might notice is that these are more industrialized nations, maybe places that have more access to vaccines, antibiotics. Maybe they use a lot more antimicrobials in their home, right, than some of these other places with lower disease risk, suggesting maybe we really have lost touch with some microbes in these places, right, that's disease causative. And so to understand diabetes a little bit more, I'm going to teach you a little bit about how the physiology of the pancreas and diabetes works. So this is a normal human, and regulating diabetes is a disease where you can no longer appropriately regulate your blood sugar, okay? And so glucose is an interchangeable term for sugar, all right? And so your pancreas is this leaf-shaped organ that kind of sits behind your stomach above your intestine. And there are specialized clusters of cells throughout your pancreas, shown here, called islets of Langerhans. And these clusters or groups of cells have hormone-secreting cells in them that help you regulate that blood sugar. The cell type that my lab is most interested in, which makes up the majority of these cells, is called the beta cell. And it produces the hormone insulin, okay? So insulin's shown here by these little blue triangles. And every time you eat something, your blood fills with sugar from that meal. Your beta cells sense that sugar, and they secrete insulin into your bloodstream. Insulin then travels to cells throughout your body, like this muscle cell, right? We're all in Colorado. We're all pretty active. It's a big deal to know what type of carbohydrate source you need that's best for your blood sugar. But insulin, without insulin binding to these insulin receptors on those cells, your muscles would have no way of absorbing the sugar from their bloodstream, okay? So that's how this process works in a healthy individual. But what about someone with diabetes, right? So there's two types of diabetes. Type 2 diabetes is by far the most common form of this disease, all right? And type 2 diabetes happens because we lose these insulin receptors, all right? They stop functioning, which makes us insulin resistant. It means that insulin is still made, and your beta cells actually end up making extra of it to try and overcome this insulin resistance. But the receptor is no longer active, and so it can't tell the cell to absorb sugar, okay? Now, type 1 diabetes is the other form of diabetes, and it's kind of the opposite. It's sort of the flip side of this. There are fewer individuals on that map I showed you with type 1 diabetes, but they have the opposite problem where they can no longer make insulin. And this happens because these little purple cells, your immune cells, something goes wrong where they misrecognize your beta cells as non-self and they kill them off, all right? And this means that you can no longer produce your own supplies of insulin, and you have the same problem where you can no longer absorb sugar from the blood, all right? And type 1 diabetes is going to be the main focus of my work, so that's what we're going to continue to talk about. And so the only way to treat type 1 diabetes is by injecting yourself with insulin, all right? Insulin that we make in the lab and then you buy and inject, all right? And so there's no cure for type 1 diabetes. And we've been using insulin to treat this disease since the 1920s when it was originally discovered. So over 100 years now, still no cure. And type 1 diabetes, importantly, is a childhood onset disease. So that's why I have this kiddo in the background here to illustrate this. The majority of individuals who are diagnosed with T1D, they're all diagnosed around the age of 3. So imagine having a child that young that you have to teach this really complex biological process to, and they now have to regulate their blood sugar for the rest of their life to survive. That sucks. So how do we come up with a better solution for this disease? Well, it turns out that type 1 diabetes is genetic, so you have to have the genetic mutations in order to develop it. And these genetic risk factors are either found in the form of mutations in that person's immune system or mutations in that person's ability to make insulin, okay? But the wild thing about type 1 diabetes is that you can have the genetic predisposition for this disease and still not get it. You can actually not have the disease even though you have the genetics, which means that there's a really big component of one's environment that's influencing the onset of this disease, all right? So in addition to these genetic risk factors, there are also a slew of environmental factors that drive type 1 diabetes. And these include things like diet, whether a child is breastfed or born cesarean or naturally, the use of antibiotics and antifungals like we've been kind of talking about all night, as well as early life viral infections. So as a microbiota scientist, when I look at these risk factors, the first thing that stands out to me is that almost all of these things, everything except insulin mutations, these are things that can impact the composition of your microbiome, how many species you might actually have in there, all right? So this is very striking to a lot of microbiome scientists around the world. And many of us have been asking, are microbes important modulators of type 1 diabetes then, right? And so to ask and understand this question, we have to also know something about microbiome development, because remember, type 1 diabetes is an early onset disease. So what happens in our early life microbiome? It turns out that infants and children have a really unique microbiota. The microbiota that you all had as babies, completely different from the one that you're sitting with right now in this room, okay? And so we're born essentially sterile for the most part, and then we start gaining a few microbes, and the number of species that we gain throughout our life grows and grows and grows. And we call this diversification of the microbiota, all right? So just like you develop and change, your microbiota develops and changes too. And this is especially important because we've also sequenced the microbiota of young children who are genetically predisposed to type 1 diabetes and asked, how does their microbiota diversify, right? How do they build species number? And so on the bottom is the age of kids, and we've got a little arrow here showing the average diagnosis of type 1 diabetes. And if we graph the diversification of a kid's microbiome, we can see that it grows and grows for a little while, and then it tops off and actually starts to decline right before an individual's diagnosed. If we look at the kids who don't get diagnosed, their diversity continues to climb and grow much, much higher than kids who are diagnosed with T1D, which means that reduced microbial species could be a factor in actual onset of this disease, all right? And so microbiota development coincides with a lot of developmental processes, right? This infant looks completely different from this young child. We grow and develop. So one of the most striking developmental processes to me that co-occurs with development and diversification of your microbiota is actually the development of beta cells in your pancreas, those cells that are secreting insulin and are really important for regulating your blood sugar. So if we graph the number of beta cells in the pancreas based on how old you are, we get something that looks like this blue line, okay? So this is an approximate estimate. When you're born, you have some beta cells, and then the number of these cells grows and grows and grows, weirdly until about the age of 3. So that number kind of keeps coming back. And then these cells kind of stop growing, and whatever you made early in life is what you have for the rest of life, okay? So there's some type of green light signal telling your beta cells to grow and divide early on in life. And sometime around the age of 3, something stops that signal, and that's how many beta cells you have. And so this is an example of a critical window of development. There are lots of cell types in your body that can only develop during certain times. Beta cells are one example of that. And so the fact that these, growing beta cells are really coinciding almost perfectly with the time when your microbiome is developing and the time when that development is really important for T1D onset led to my hypothesis, which is that maybe there are microbes found only early in life that influence the growth of beta cells in your pancreas. So that's my hypothesis. So it's really hard to study the microbiome. I've been sitting here telling you all night how complex it is, all of these interesting facts about it. So how do we actually study it as scientists? What are the tools that I need to answer this question? So we have some very awesome model organisms that my lab uses in order to study the microbiome. And these are actually germ-free or sterile mice. So these are mice that have no microbes associated with them at all. So once we have these sterile mice, we can give them whatever microbial community we want and see how that community impacts that mouse's biology. How does that microbe impact this mouse's beta cells, okay? So we've got a community that's a normal microbiota. It's got all those things in it that we talked about, from bacteria to fungi and viruses. And then we can have a germ-free mouse that has nothing at all in there. But what's so cool about this is that once there's nothing, we can add back individual bacteria and ask, how does this one microbe impact my process of interest? Okay, so how do we keep these mice sterile? How do we do that? There's microbes everywhere, all over, inside of you, in our room right now. We actually keep these mice in a vinyl bubble. Pretty basic. We just have this physical barrier, and inside of this bubble, everything is completely clean, microbe-free. There's nothing in there. And we have these mice in this facility here at BioFrontiers, and they're a really cool and powerful model to continue studying this system. So we know how we study the microbiota. We need big plastic bubbles. How do we study the pancreas? How do we know how many beta cells are in the pancreas, okay? Well, we actually take the pancreas out of the mouse and count the beta cells. How many are there? And this is a cross-section of a whole mouse pancreas, all right? Everything in purple is a stain that we use to identify all the cells in the pancreas. And if you're squinting really, really hard at this image, hopefully you might be able to see these tiny brown dots, all right? So if we blow this up, all of these little brown dots, those are those islets of Langerhans that I told you about. And everywhere that's brown, those are beta cells secreting insulin. So we literally just count all of the area that these insulin cells cover, and we come up with this measurement called beta cell mass, okay? And that's how we know how many beta cells grow in response to whatever microbe we give the mice. So if we do this experiment, if we look at beta cells in the pancreas of a mouse with a normal microbiota, hopefully you can see that there's lots of these little brown islets of Langerhans throughout there, okay? Plenty of cells to make insulin for this mouse. But if we look at the same pancreas in a germ-free animal, no microbes, there are many fewer of those brown islets of Langerhans. So microbes are important for stimulating beta cell growth, which is really, really cool. We can replicate this experiment by treating our mice with antibiotics. So instead of taking those mice out of that plastic bubble, we just give them those antibiotics to wipe out their bacteria, which you and I have taken before, which we're giving kiddos, right? The same thing happens. There's many fewer of these little brown dots. So microbes are really important for beta cell numbers. So how do we tell if early life microbes in particular are important for beta cell numbers, right? We have to get a little creative in our experimental design. So we have designed 2 experiments that I'm going to tell you about tonight that helped us to test this question. So the first one is that we deplete the microbiota with antibiotics and antifungals at different times of the mouse's life, all right? So we take the microbe away and ask, if we take it away early in life, did it do anything? The opposite is we can add the microbe back. So if we give the microbe back to germ-free animals at a certain time, is it going to restore function? Okay, so here's that experiment laid out. Here's the timeline of the mouse's life. And everywhere that you see these colored bars is a time window when we gave antibiotics to take the microbiota away. All right, so we did it at different time points throughout the mouse's development. And importantly, these time points coincide pretty well developmentally with similar time points in a human's life. Okay, so we have this first one, which is actually in utero, so before birth. This next one is during infancy. And then this final window is like around the age of weaning. Okay, so about 1 year of life in a human. So if we look at beta cell mass measurement, this is what it looks like if we graph all of those blobs, all of those brown dots. So we get that beta cell mass. And again, a normal mouse has lots of beta cells, whereas a mouse that's germ-free or on antibiotics doesn't have as many. And each one of these little dots represents one mouse pancreas that we looked at. So we looked at a lot of mice. And you might see that there's a lot of variability in how many, there's a lot of variability in the beta cell mass between mice. And this just represents some of the natural biological variability that we as scientists have to deal with. We can't actually control every little thing. But overall, there's more beta cells in this mouse with a normal microbiota. So what about these time points? What if we take away the microbiota early in life? So if we take away the microbiota in utero, we see no effect on beta cell mass. Similarly, if we take away the microbiota early in mouse infancy, no effect on beta cell mass. Their beta cells are fine. But if we take away the microbiota just before weaning, oh my gosh, it looks like these mice have been germ-free their whole lives, right? So as a scientist, this tells me that there's a really critical window of time where microbes are doing important things for beta cell growth just prior to weaning of the animal. So this is wild. Microbes are stimulating development only at certain times in early life, all right? So good scientists, we design multiple experiments to test the same question so that we can prove it in different ways. So our second experiment that we talked about was to now add the microbes back at different times, right? So now we're gonna validate what we saw in our last experiment. And so this is the layout. We're building on top of that last time frame I showed you. And remember, here's our critical window in this blue bar. We're either gonna add our microbes back after that window ends, so the animal is germ-free all the way until the end of the window, or we add the microbes back before the window starts, right? So we're gonna keep them germ-free until here. And we'll compare both of these treatments with animals that are always germ-free. So what does that look like? Well, it validates what we saw in the first experiment, right? If we add the microbes before the window starts, we have a normal beta cell mass. But if we wait just 10 days, we lose beta cell mass, right? So these mice look like they've been germ-free their whole lives, but we just waited 10 days longer to give them microbes, which means that there's a very fine time frame in which the host is primed to receive that signal. It's really wild. So again, this time frame sort of coincides with what we would predict in a human to be around 1 year of age or around weaning, okay? So then does the same thing happen in humans? We've looked at mice. Clearly mice have this issue, but what about our kids? Are they also enriched with certain microbes at a certain time that's promoting development in their pancreas? It's really hard to study humans though. I can't study them as well as I can study mice because they eat whatever they want. Try controlling a 2-year-old's diet, right? Also, their genetics are all completely different. And I can't just willy-nilly go around peeking into people's pancreases, unfortunately. So how do we test this of whether the microbiome alone is a factor in humans, right? We have to, again, get really creative as scientists. And so what we do is we can actually collect just the microbiota, which, yes, is poop samples, okay? So we can take poop samples from kids at each of these different age groups and give them to our germ-free mice. And now our germ-free mice have a human microbiota. It's really cool. So we can ask, do these human microbes stimulate mouse beta cell development? Okay, and this works because mice allow us to control them. We can feed them all the same thing and they're happy, and they all have the same genetics, and they don't complain too much when I take their pancreas, okay? So the other thing I want to point out about the way we designed this experiment is that we didn't just get poop samples from any kid. We got poop samples from kids who are genetically predisposed to type 1 diabetes. And we got these from this really amazing global study called TEDdy, which is the best acronym I've ever heard, which stands for the Environmental Determinants of Diabetes in the Young. And importantly, we got samples from kids who were genetically predisposed, but were never diagnosed. So some of those kids who stayed healthy, which also allows us to control for the human genetics shaping these microbes, which is really powerful, okay? So if we take all these samples and we put them into our mice, this is what our beta cell mass looks like. So again, each dot is a mouse that received a microbiome from a human, and they received microbiomes from all of these different age groups, all right? This is the age of the human donor. And what should stand out to you is that donors between the age of 7 months and 1 year had really good stimulation of beta cells in mice, which suggests there's a really cool window in humans too, where microbes are enriched that can actually do and stimulate this biological process. And not only does this time frame match what we predicted from our mouse model, but it also matches predictions that people have taken from human cadaver tissue where they've guessed when beta cells are the most proliferative. So this is really encouraging and exciting to us that this same process could be happening in humans. But how do we know which samples or which microbes in those samples are actually stimulating that beta cell growth, right? I just told you earlier there's 500 to 5,000 different species in one sample. How do we figure out which one of those it is? So we turn back to our sequencing approach, right? And that tells us who's present and how many are present. And then we can develop candidates from that we can actually isolate away from the rest of the community, allowing us to do experiments where we just add that one candidate microbe back to our germ-free animals and ask, does that microbe stimulate beta cell growth? And it's really cool because some of these microbes do stimulate beta cell growth. So we found a couple. They're labeled here. One is an E. coli strain. One is an Enterococcus gallinarum strain. But both of these microbes on their own promote beta cell mass. Not every microbial candidate that we picked out does this. So we have one lactobacillus species down here that does nothing to beta cells. This is important though because it's not all the microbes in these kids' guts. It's only a handful of them, all right? And in addition to bacteria, we found some of those microscopic yeast I mentioned of the Candida species that I kind of previewed. So we have this Dublinensis species that can promote beta cells, but we have another Candida species that can't do this process, right? So again, not all microbes can do this. All right. So some of you might have grimaced a little when I said that E. coli was one of the things that did this, or Enterococcus, right? How much lettuce have you avoided when there's an E. coli outbreak with lettuce, right? So again, these microbes that we're finding in these infants, it's unclear whether they're pathogens or commensals, and this gets back to context. So E. coli, Enterococcus, Candida, all of these things can be pathogens. They do things like make us sick when we eat bad lettuce, cause life-threatening infections, and those pesky localized infections that we all have to deal with, right? But what's wild is that all of these species are really common members of the infant gut microbiome. All kids have these. But they're not causing disease in all kids, right? They're providing important stimuli for development. And us understanding the context of when they're diseased and when they're healthy is an important work that we have to continue so that we're not giving kids potentially dangerous pathogens to fight diabetes, right? So how do we get around that? How do we treat kids with the powers of these microbes without getting them really sick, right? And the answer is that we actually have to identify the microbial product that's responsible for stimulating beta cell development. And so this is a biochemical image of a protein that we identified from some of our microbes that on its own promotes that beta cell developmental process. So now we can treat with just this protein. And we found this protein because it's secreted by some microbes in the gut in order to compete with other microbes in the gut. And it's just, the microbe is doing its own thing, but some of this protein scoots its way to the pancreas and actually stimulates beta cell development. So wouldn't it be cool if instead of treating with E. coli, we could just treat with this protein, right, in order to stimulate beta cell development? So that's the goal of my lab moving forward, is to try and navigate the nuance of a pathogen and a commensal, and how can we harness the good parts of a pathogen to make them helpful for treating really important diseases, okay? So how would we actually use this to treat disease? Well, again, we know that there's certain microbes that are stimulating beta cell proliferation around this time frame. What if that kid has to go on broad spectrum antibiotics right at that important window, right? I have never met a parent who has a kid who hasn't had to go on antibiotics at some time, right? It's very important that we don't avoid antibiotics to treat dangerous infections, but this would predict that we would lose some of those important signals that stimulate beta cell growth, and then that person would potentially develop less beta cells that they would have for the rest of their life, maybe making them more susceptible to diabetes, all right? So our goal in our lab is to figure out what are the microbial components that we could co-treat with antibiotics with, right? How could we supplement back things that might be lost? We don't want to tell people not to take their antibiotics because sometimes they really are necessary, especially in early life when kids need help fighting those infections, okay? So could we bolster early life beta cell mass and prevent some of these losses? And then lastly, we also think about harnessing these things not only to treat kids early on, but adults who have type 1 diabetes, right? Can we regrow their beta cell mass in order to cure disease? So that's another way that we're thinking about this problem. Okay, so I've just spewed at you for an hour, all of the nuance and complexity of your microbiome. So how do you navigate your microbiome? Many of you may have noticed I'm expecting, right? So I'm gonna have to navigate my own child's microbiome in the future. So what am I gonna do? And the advice I would give you is to trust your gut, okay? Embrace your microbes, but not at the expense of contracting potentially known pathogens. Maybe sometimes don't use Clorox on your kitchen, right? Let your kid go outside and run around in the dirt. I'm definitely gonna let my dog lick my kid. I'm not gonna worry about it, okay? But know your own risk tolerance. Everyone is different, all right? But hopefully you now understand some of the importance of a diverse microbiome. And if you want to learn more about my work or learn more about some of the science behind it, you can go to my website, which is hillabmicro.bio. And I can also recommend some reading material for a general audience from experts in the microbiome field. Ed Yong is a really good scientific writer for The Atlantic who put out this great book. And then the rest of these books are by leaders in the microbiome field. I mentioned Martin Blaser. Justin and Erica Sonnenberg are a husband-wife team at Stanford who are leaders in this field. And then Brett Finlay is another leader at University of British Columbia. I wanted to point his work out because his daughter, Jessica, is an assistant professor here at the University of Colorado Boulder. She's in the geography department studying global health patterns. So some more ways that Boulder's been involved in translating information about the gut microbiota. All right, so the last thing I'll do is just highlight the amazing people in my lab. This is what microbiota scientists look like who are doing all of this work. And then obviously it's been awesome being a part of being a new member of the BioFrontiers Institute and then all of the organizations that have supported or continue to support this work. So that's all I got.
Thank you, Jennifer. And if anybody has questions, we have time for a few questions. Please raise your hand and we'll run mics to you. Back there.
So you mentioned that the protein that you found inhibited other microbial communities within in the gut. Is there a fear of treating with that causing an imbalance in the microbiome since you've now favored one over another?
That is such a good catch and a great question. So the really cool thing about that protein is that for some reason, beta cells are incredibly sensitive to it. So we could imagine giving someone a really low dose of that protein and their beta cells would respond without impacting the rest of their microbiota. Yeah, good catch, great question. Hi.
So, given that bacteria are constantly talking to each other, does it surprise you at all? I mean, it surprised me that you saw a response from adding a single species back.
Yeah, it was really surprising to us, especially given the complexity of the whole microbiome. I actually anticipated that there'd be no way that we would see one species having an effect. But we actually see the opposite, where there are lots of individual species that have this effect, right? Which tells me that the host or the animal has evolved ways to listen to lots of different microbes, and that this isn't about the bacteria stimulating a process in the host. This is about the host learning to listen to bacteria. And if you're going to hang your hat on a bacterial signal for the development of a really important cell type, you want to be able to listen in to multiple different types of bacteria. So rather than all the bacteria doing it, what we're seeing is there's just a lot of individuals that can do it, which is kind of cool. Yeah.
All through the same molecular pathway?
Different molecular pathways. Yeah, it's really wild.
Got one over here. As you look for hits of microbial products, have you seen anything that's known to be immunomodulatory? On like macrophages or anything like that?
Yes, we have. So there are microbes that actually modulate macrophages in order to stimulate some of these processes. Yeah, yep.
So I'm wondering whether either young children or adults that get high doses of broad spectrum antibiotics have been found to develop diabetes when they didn't have it before.
Yes, there's a general trend with antibiotic use and higher incidence of T1D, but it becomes really difficult to track individually back whether an individual's exposure to antibiotics was the cause of their disease. And that's because there's a wide variability in antibiotic type and use and maybe timing at which they took it. So one thing we're trying to do in our lab is mine a lot of this metadata from medical clinics that are out there and find out if anybody who took antibiotics from 7 to 12 months of age, did that actually put them at higher risk? And so we think this time frame that we found might actually help in solving that question. Yeah.
A couple down in this row. I think I saw her hand.
I was wondering, since you found this protein that will stimulate the development of the beta cells, is there any danger of when you apply it, it would cause an overproduction of the beta cells, or that red light is still there?
I love this question. Yeah, so a whole other aspect of my lab's work is what if you take these pro-proliferative signals out of context, right? And we think about this a lot when we think about other diseases like cancer. And so it turns out that pancreatic cancers, which are an adult onset disease, those individuals tend to have a weird recurrence or an enrichment of microbes that are more prevalent early in life. And so there's a whole other arm of my lab looking at if you take early life microbial signals out of context, will they cause cancer later on? Yeah, because it'd be very important to know if we want to treat these to adults with type 1 diabetes, are we also putting them at risk for cancer? Yeah, it's a great question.
You listed several things about the increasingly clean environment that we live in, but one of them was vaccinations. And I'm curious, have you looked at can you speak a little bit further to that in terms of type 1 diabetes?
There is no trend of vaccination with type 1 diabetes. Yes.
Thank you for clarifying that.
Thank you for asking that question. Yes, yep.
Does ivermectin? No.
That's a virus.
Tell us how AI is affecting your work or will affect your work or threaten your work.
Great question. I am behind the times for AI, unfortunately, but I think if anything, it's going to help us, right? And so one way that we're thinking about using AI is I showed you this picture of this protein, right? And so this is a protein that is really good at promoting beta cell development. So one way that we're trying to use AI is to screen lots and lots of microbial genomes for similar proteins to see if we can find more that might have the same impact. So I think if anything, it's a really, really helpful tool for us. I don't see any way that it's threatening our work at the moment, but it's definitely something that we're continually thinking about. Yeah, it's a great question.
Sorry, I'm curious about, the mechanisms that open and close the window in human development for the production of beta cells. Do you foresee a future where we'll understand enough that we might be able to reopen that window at later stages?
That is one of the big questions that we're asking is what are the rules for that window? And right now, all we know is that it's driven by the enrichment of certain microbes. But what we don't know is whether the host has another regulatory mechanism for sensing that window, right? And one of the problems with beta cells is that it's really, really hard to get adult beta cells to proliferate, which is why it's so hard to treat type 1 diabetes. And so I suspect that the beta cell might only be receptive to those signals for a certain amount of time. And so we're also curious, how could we control the beta cell to be receptive to those signals all the time, right? Or when we want them to.
Thank you.
Yeah.
All right, maybe one more question over here.
I didn't know I was going to be last.
So a lot of pressure, right?
Yeah, I know. Anyone else? No. So going off of that window, how consistent are the different species of microbes from person to person in that window? Is it always the same, or not always, but consistently a similar microbes that are in that window? Or someone that, is, in Iceland versus the US versus, what's going on?
Yeah, such a good question, Skip. Yeah, great question. So I have a grad student in the lab who's trying to answer that. And so that study that I mentioned, the TEDdy study, has samples from kids all over the world, all over different geographies in the US and Europe and Australia. And he's asking, if I look at that window across all these different populations, are the microbes in there the same? Or is that window potentially different depending on where you are, right? And